Good afternoon, everybody. My name's Doug Brown. I'm the Chief Executive of the BSI, and we would very much like to welcome you to the second in our series of Connect on Coronavirus, the Expert Hub. Thank you very much for joining us. We had over 2,000 people sign up for this webinar, which is an absolutely amazing response. So we had our first one last week and even more people signing into this one this week. So thank you to all of you who were spreading the word. We really want to be offering this platform out to the research community at this point in time to try and support all of you to quench your thirst for new immunology understanding, but also to share that understanding, that learning, to stimulate the discussion within the scientific community, which is really going to help us move forward as fast as we can. As I said last week, our viewerage today is global as well. So we have people from North America, Europe, again, the Middle East, over out in Australia and New Zealand as well. And this is just the second one of a number that we will be running over the next few weeks. So please do stay tuned for the announcement of more webinars over the next week or so. For those of you that don't know, though I'm sure most of you do, the British Society for Immunology is here to support all of you, the immunology community and wider. Please do find out more about what we do if you don't already know by visiting our website at www.immunology.org. These webinars are just one thing that we are doing in response to the ongoing pandemic. There's lots of other initiatives that we are delivering and new ones we will be launching, and the information is all on our website and via Twitter, so please do follow us. For today, it's my absolute pleasure to be introducing our speaker. We have Professor Danny Altmann, who is Professor of Immunology at Imperial College London, and I will hand over to him in a few moments to begin his talk. Logistically, as of last time, we have asked Danny to speak for around 30 minutes or so, and then we have 10 or 15 minutes for questions afterwards. It's now time to hear from our speaker, and Danny today is talking about the emerging lessons about immunity for COVID-19. So lots of new research coming out. Danny has been trawling the archives and will be giving us his insights on what has been coming through. So Danny, we very much look forward to it. It's over to you.
So thank you very much for joining this webinar. As you heard, I'm an immunologist at Imperial College, and for the last 20 years or so, we've held a weekly journals club. So for those of you who aren't academics or medics, a journals club is a bit like a book club, except without the white wine. And the idea is that you meet once a week and you dissect a new publication and try and embed the message from it in your long-term memory. And over the last month or so, I've been kind of quite obsessively trying to read all the papers coming out about the immunology of COVID-19. So today I'm going to cheat a little bit, and instead of presenting the message from one paper, I'm going to give you snippets from 5 or 6 that I thought were quite good and quite important. You'll see that many of the papers come from China and from Wuhan in particular, and that's because I think there was a fairly heroic effort by clinician scientists in China to collect some fairly hard lab data in the face of their developing pandemic. So if I go to the first paper, hang on, you can see that this is a paper from China. It's of a type that's now come out from many centers and many cohort studies. It's telling the story of some of the basics of the virology and the immunology of the evolving infection. So I thought it would be good to sort of stand back for a moment and ask what are the kind of questions that immunologists try to sort out for any emerging infection, because they're all different and the details kind of matter. So which are the viral antigens that the host immune response targets to resolve infection. Are we just looking at neutralizing antibody against the spike antigens? So I guess everybody will have heard a lot about the spike antigen. It's definitely a key, or possibly the key target for immunity, because it's that spike on the surface of the virion that it uses to attach to the host receptor. And we certainly know that antibodies against spike that are capable of neutralizing can block infection. So that's clearly a key place to start looking. But is that antibody the only or the key correlate of protection, or do we need to understand more? And you'll more about that in a moment. The other part of the immune response, or the other key part of the immune response, T-cell immunity, is clearly heavily involved because, as you'll see in a minute, it's desperately impaired in people who do badly. So we need to know to what extent T-cell responses are a correlate of protection. If that's the case, are we mainly worrying about CD4 T cells, the ones that help for antibody production, or are we meant to be worrying about CD8 cells, the ones that kill virally infected cells, or are we needing to think about both? So overall, what does long-term protective immunity look like to this virus? Is it just the antibody response, or do we need to know more? So if we look at the next slide, this is the study from the Chinese cohort. So in this, as you can see, they looked at 67 hospitalized patients between the end of January and the beginning of February, and collected clinical specimens for PCR. And some of the detail is quite interesting. Yeah. So if you look at the results from the swabs, many of them were positive for virus out to day 38 after hospitalization, which is actually quite a long time if you think about it. And the median day for PCR positivity was day 12 after hospitalization. Something that's been reported quite widely, but I'm not sure if it's been taken on board, is that many of these people were PCR positive for virus in their stools, in their poo. So some of them quite far out to day 26 after hospitalization, with a median time of 18 days. And I know there's been some discussion in the literature about whether that's definitely measuring real life infectious virus, or whether it's just PCR amplifiable RNA, but definitely worth thinking about. And there's even a sizable minority that have PCR positive virus in their urine and in the blood, in their plasma. A common observation is that there was very prolonged enhanced viral shedding in the more severe patients. If they were coughing, they were probably shedding virus because the two were correlated, although fever wasn't. And antibodies were seen from day 7 in terms of IgM, and from day 10 in terms of IgG. Something again that comes out from many of the studies that at first glance is kind of counterintuitive, is that the antibodies appear earlier and go higher in the more severe patients than the non-severe patients. Yeah, so making a big antibody response wasn't a marker of doing well, it was a marker of doing badly. It said you had more virus on board with more antigens stimulating more antibody, and the people actually with lower antibody responses got rid of the infection faster and cleared it faster, presumably because they had less in the first place. So to home in on some of that detail in one of their tables, it's kind of informative. So if you look at this right-hand column here, this is just the PCR result, and it says that even in people who've got barn door obvious COVID infection, only 97% of them are ever PCR positive, presumably because the disease is heterogeneous in different people, and some people went negative at different times and never got caught as positive by the PCR. So 3% of the people would never have been scored in your stats as PCR positive. And so if you didn't get them by the PCR, would you have got them by the antibody test? So let's look at this column in the middle here at the IgG. And the answer again is that even people who've been hospitalized with severe infection, antibody positivity never reaches 90%. So 10% of the people who have full-blown infection would have been scored as negative by an antibody test. either because it was too low and beyond detection for their assay, or because they were perhaps people who were very, very severe cases who were going to die, who simply made no detectable antibody, or because maybe you should have been measuring some other parameter of immunity, like, for example, a T-cell response. So if we have a look at the next study, again from China, from Wuhan, it's quite a kind of heroic effort in the midst of that flow through of very severe patients to have collected a cohort of 522 hospitalized cases, about 10% of them going to intensive care, and do some fairly sophisticated T-cell immunology by flow cytometry. And that's what they did. And I want to talk for a moment about some of the take-home messages from that. You'll see at the bottom here that one of the things they look at, which is quite interesting, and we'll come back to it, is whether the T cells that those patients have are exhausted T cells. We'll talk more about that in a moment. So the take-home message from their study is that the severe cases who are hospitalized have a desperately low T cell count in their blood. Both their CD4 and CD8 compartments are very much reduced. And that's especially seen in people who are elderly and or those who need intensive care admission. And the very lowest counts are very much correlated with a fatal outcome. Okay, so that's part of the immune response that's very much shut down and impaired in a fairly dangerous way. On the other hand, some parts of the immune response are souped up. So the patients who did the worst had the highest levels in their serum of the cytokines IL-6, IL-10, and TNF. And we know that both those two facts are somehow related to disease outcome, because as the people recovered, it was associated with those cytokines going down and T cell numbers being restored. So if you can restore those T cell numbers back to normal within a matter of days, it kind of suggests, I think, that the T cell counts weren't low because the T cells were killed. It's because they'd been sequestered somewhere else and weren't detectable in the blood. And then they make this additional point, which is quite interesting, which is that the T cells that those people did have on board had this exhausted phenotype. So the name is as it suggests, and it comes particularly out of the tumor immunology literature, as in the Nobel Prize a couple of years ago, which said that the reason that some cancer patients don't make a good immune response to their tumors is that the T cells that get into the tumors express these markers, things like PD-1, which correlate with a kind of exhausted phenotype where they don't have good effector function and can't kill the tumors. But if you can block the PD-1 function with an antibody, you can restore that function and get them to do their stuff again. So when this group looked at those markers in these patients, you can see in this graph here that the problem for them is not just that they've got a very depleted supply of T cells, but that the ones they have these markers of exhaustion and can't do their job properly. And that's all the more so if they're the people who go to intensive care. Okay. So I mentioned the very high IL-6 in the serum of people who do badly. And that's an observation I think has come now out of many different studies. And obviously everybody has an interest in trying to discover biomarkers that might help you to manage patients better and to spot ahead of time who's going to be at risk of deteriorating and needing ICU. And maybe if you could spot them before it's too late, you could do more for them sooner, which I guess is what biomarker discovery is all about. And this is quite a good paper showing that it's probably worth in those people trying to find a way to monitor IL-6 in a hospital setting, because that, with a massively strong p-value, predicts which people are headed for mechanical ventilation. Many people will also obviously be aware that IL-6 is a target for a therapeutic monoclonal antibody called tocilizumab, which is in clinical use and licensed. And I've seen some clinical trials trying to use it in these patients. I'd be interested to hear what anybody else thinks, but I've seen some data that looked quite promising and some that look quite equivocal. So I think it's like many of the other treatments, it's a matter of watch this space. I wanted then to mention another paper, one that Ani Akbar mentioned in this webinar last week. So now we go from a paper of 522 people to a paper in Nature Medicine, which is a case report of one person. But I think she's quite a helpful, illuminating person for understanding what's going on. She's a 47-year-old lady who flew from Wuhan to Melbourne, Australia, and clearly had been sort of incubating the infection for a while, because if you have a look here, after she arrives in Australia, she starts to develop symptoms over a period of days, and 11 days after arriving in Australia, presents to A&E with a high fever. She's not one of those very severe patients like the ones we were just talking about, but on the day that she presents to hospital, her swab is positive by PCR. But have a look at the data from her, because it's kind of helpful. So 3 days later, if you'd done the PCR only on day 7, you'd have scored her as negative because she's no longer PCR positive. If you looked at her feces on day 6, you'd have scored her as PCR positive because you can detect virus there. But by day 7, after she's gone into hospital, there's nothing left to detect. What you can detect, though, is antibody in quite a sort of fancy antibody neutralization assay here. She's in her blood. She's got IgM and then IgG antibodies to the virus. Okay, so in the people we were talking about just now, you can't find T cells. In this lady who's much less severe, you can find emergence and rapid expansion of very activated T cells, especially CD8 cells, that seem to correlate with and relate to resolution of her symptoms. So some kind of marker perhaps of successful functional immunity that enabled her to go home from hospital. So should we be adding some kind of T cell analysis to form some part of the arsenal for immune monitoring for this disease, which obviously wouldn't be amenable to home testing or GP testing? And this is a tweet that came up yesterday from Bertoletti's lab in Singapore, showing that they've developed precisely that kind of assay, which flags up the patients with immune memory, and interestingly does it for a different antigen from the virus, not the spike antigen that we're looking at in the antibody response. So I think, again, it's another kind of matter of watch this space. So you'll have gathered there's a kind of emerging theme from these papers that says that it's incredibly important and incredibly non-trivial that as time goes by, we need to work out how to evaluate long-term protection, whether it's by vaccines or by natural infection, and understand something that immunologists angst about enormously: what are the true correlates of protection? And if we knew what they were, how much is enough? Is it just the titer of neutralizing antibody? Is it the CD4 and or the CD8 T cell response? And these are really, really key questions that you'd want to have answers to. For example, if you were trying to decide which vaccines to invest money in and develop and go with and license. So what would protective immunity look like, and how can we work out who has it? Clearly, an enormously pressing question at the moment that many, many people are talking about. So this is the slide where I try and articulate what we mean.
It's 12 hours.
I try and articulate what we mean by the gap in seroprevalence data. So when I think about it, I'm not thinking about it purely in terms of the way that it's sometimes been presented in the press, in terms of trying to work out who's got antibodies on board so that we can work out who could go back to work. So I think it's kind of bigger and more complex than that. It's trying to work out where we have this disease, who has this disease, how many have the disease, whether we're heading towards herd immunity and what that immunity looks like. So to try and explain the point, on the left-hand side of this slide is a table from a paper that came out of Imperial College last week, which tries to model for different European countries the percentage of the population that might have been infected. And the estimate that the study makes is that for the United Kingdom, if you have a look at the bottom of the table, 2.7% might have been infected so far. So if you try and translate that into numbers, the modeling data says that the UK has about 1.6 million people who've so far been infected. That obviously is an enormously different number from the numbers that are quoted every day as having been PCR tested, because the number of PCR tests is limited. So that records the daily stats the last time I looked as 47,000 cases. So if you think about the difference between 47,000 and 1.6 million, that suggests that we pick up about 1 in 35 of all the cases, and the 34 others that we don't pick up are either unwell at home, untested and unrecorded, or they're asymptomatic and didn't mind. So thus the enormous pressure to have a different kind of test to work out what's been going on. So developing tests to look at antibodies is a thing that immunologists have been doing for many decades. And there's a number of things that you need to think about when you're trying to work out the antibodies that are in your blood sample. As I've mentioned, there are many antigens in your virus, and you need to decide what you want to measure antibody to. So most people are looking at antibody to the spike antigen, which from all the things we've said just now seems a pretty good place to start looking. Do you want or need a simple yes/no answer, or do you need some kind of more nuanced quantification? Do you want the kind of lab-based test that a technician or a scientist can do in a laboratory setting with fancy equipment, or do you want a point-of-care test that anybody could do on a pinprick of blood? The latter obviously gives you a much simpler, more accessible, high-throughput answer, but demands loads of technical development and manufacturing refinement to develop a foolproof module of a type that would normally be in development for a couple of years, not a couple of weeks like the timescale we've allowed ourselves here. You've got to ask yourself whether it matters to you to truly be able to measure functional antibody that's actually protecting against the virus, neutralizing antibody, or do you just want to know if somebody's seen the virus or not? If you don't have any handle on neutralizing virus, you might be kidding yourself thinking people are neutralizing antibody. You might be kidding yourself thinking people are immune when they're not. And these are choices that have quite big implications for the quality and the quantity of data you can get, the cost of it, the speed of developing it, the materials you need, the validation, and so on and so on. And for people who want to learn more about this, I'd suggest to have a look at the blog out of the University of Oxford that John Bell posted yesterday that talks about the process that the UK has gone through in trying to validate its antibody protocols. So many countries and communities have done this in many different ways. This is a little editorial from Science talking about the efforts out of New York, and they refer to, and this man on the right here, who's called Florian Kramer, who's a professor of virology in New York, and he was far ahead of the curve. Some months ago, he turned over his lab to a kind of cottage industry to develop recombinant spike antigen and a validated protocol for measuring antibodies, and he's sent that out as a kind of gesture to the community, to anybody who's requested it, and many thousands of people have been tested in this way. And there's a lot of data in the bag from that. But obviously that's a lab-based assay, and kind of at the other end of the spectrum, you have the kind of thing that all countries are trying to develop and purchase and get their hands on, which is an optimized and validated point-of-care test that could really tell you in the community with high throughput how many people have antibodies. So how can we go about trying to get a handle on how many people out there have antibodies and have seen this virus? Are there any clues? So some of you will have seen this article in the newspapers at the weekend. It comes out of Italy, And it says something rather interesting and perhaps rather positive, that there they looked at blood bank donors, and they found that 70% of people who were asymptomatic, had no idea they'd met the virus, were actually carrying antibodies to this thing. So that says antibody is already there. It's present in far more of the population than you might have guessed, which sounds kind of positive and useful. On the other hand, there's the data that I showed you at the beginning from this Chinese hospitalized cohort, saying that on the other hand, there's 10% of the people who've clearly seen the virus, who, if you try and measure their antibody responses, are this grey curve here, where they're below the limits of detection and appear not to have any antibody on board at all. So there's some people who have been infected who don't have any detectable antibody and would come out as negative in your serological screen. Are they still protected? We don't know. We need far more immune monitoring to know that. So although there's quite a steep learning curve about this coronavirus, you can get some quite good guesses about long-term immunity from the nearest relatives, SARS and MERS, because my feeling is that the general rules of protective immunity and antigens tend to follow similar trends between closely related viruses, except where you have family members that might have evolved specific mechanisms to undermine or subvert immunity. So have a look at this. This is a long-term, year and a bit longitudinal study of antibody responses of people who've been hospitalized with MERS in South Korea. And it makes the point again, so now they're doing quite a fancy antibody test to look at real-life neutralizing antibody. And it shows there are all these people down here on the bottom of the graph who had severe enough infection to be hospitalized, they had pneumonia, but they're making no antibody response to speak of. So it's the point here at the bottom of the summary. 4 out of 6 of the patients who had what they called mild disease, but did encompass pneumonia, was undetectable. So if you can't find evidence of immunity by antibody, can you find it by T cells? Well, this is another study on MERS from one of the major labs, and it makes 2 points. One is that antibody certainly is a correlate of protection by some criteria, because you can protect infected mice by transferring antibody. And the other thing to say is that some of those people who you saw before on the bottom of the graph, the green lines of undetectable antibody, have rip-roaring CD8 T cell responses against the virus, which we presume might be protective if they met it again, but without fancier studies, hard to know, for this coronavirus or any other. And the point here on the graph is the one that we made before, that having a high antibody response kind of isn't necessarily a marker of doing better. It's kind of a marker of doing worse. It says you were on ICU for more days, had more viral shedding, and met more antigen. So just to finish off in the last 2 slides, as immunologists, obviously we're all getting asked where the vaccine is and when it's coming. And all I wanted to say about that is that, as you'll all be aware, there are more than 20 candidates in preparation. And I think that's really testament to progress in immunology and vaccinology over the last several years, that there are these terrific platforms and pipelines that are kind of ready to go for precisely an eventuality like this. And most of these are safe bets to the extent that they're plausible, established technologies with plausible antigen strategies. But they're quite diverse approaches, so they'll give you very different flavors of immunity, different magnitudes of responses. Some might be one-shot vaccines, some will need a booster, some will be easy to scale up, some will be harder, some will be very heat stable, some less so. So really, important choices and sort of nuance there. 3 have so far moved from basic immunology to phase 1 testing to look at both safety and immunogenicity in volunteers. And the big bottleneck comes in how to assess which of these candidates really have efficacy, because not all the ones that can generate an immune response will necessarily be proven to protect. And even if there's a simplified or foreshortened route to licensing, there'll need to be very strong assurances about safety if these are going to be rolled out in multi-million doses. So this is, we won't dwell on this, this is a summary of the vaccines, but you can see it in New England Journal of Medicine from last week. And I'd simply finish by saying that my own personal prediction is that a handful of these will make it to a finishing line for large-scale synthesis. I find it hard to imagine that that won't happen, very much before the end of the year. The scale of this kind of synthesis from a standing start will be really mind-boggling and unprecedented and challenging before any vaccination programs can start. So, my guess would be sometime next year. And with all those different candidates, my guess is that different countries might opt for different solutions. So thank you very much. That's the end of my presentation, but I'm happy to take questions.
Thank you very much, Danny. That was a fascinating presentation and a real tour de force of what has been coming out over the last few weeks and couple of months with regards to the virus and COVID-19. As you can imagine, we've had a huge number of questions and they're still piling in now. So thank you everyone for your questions, and apologies in advance if we don't ask your question, because there's just no way we'll be able to ask every single one, but I'm trying to group them and pull them together in themes, so hopefully we'll touch on all of the key elements that are being asked. So Danny, if we could just kick off with the first question, sort of talking generally about the disease. Can you elaborate a bit more on what mechanism you think is causing the damage in the lungs of severe patients?
Yes, so I mentioned some of the cohort studies and clinical studies. There've been terrific ones. So I'd really urge people to have a look at some of the big studies, particularly in the Journal of Medicine. And this is like many severe lung viral infections, where you see people sort of rumbling on for several days, and then getting more and more impaired lung function, and some of that is inflammatory damage, and it's immune inflammatory damage, which is why I think that some of the immune cytokine markers, things like TNF and IL-6, are such biomarkers of things going badly. So some of it is immune pathology.
Great. Thank you, Danny. And we had quite a few questions about targeting IL-6 as a potential treatment option. And somebody who's joined us from Spain, thank you very much to our overseas viewers, but saying that some hospitals in Spain are using tocilizumab in patients with IL-6 serum higher than 40 picograms per mil, and they seem to be responding well. Did you want to comment on that?
Yeah, so clearly they've got far more personal expertise than I have. So I think it would be great to see people coming together and pooling their data. So I've certainly been aware of some very strong data, some less strong data. So it's going to need some trials of sufficient size and scope. My hunch from everything we've said this morning is that where it's done, as the person in Spain just said, in the context of monitoring of IL-6 levels, I think it could be really, really useful. But, it needs the data.
No, definitely. And I mean, on a similar theme, do you think serum antibodies are the best guide to immunity to respiratory viruses, or would secretory IgA be more helpful?
Yeah, no, brilliant question. So. I think immunologists feel nervous, don't we, at the current absolute, monolithic obsession with measuring spike, IgG, and peripheral blood, as if that will be the marker of whether planet Earth has immunity or not. And without doing a load more studies, we simply haven't the foggiest notion. I mean, it's in there somewhere, and it's some kind of correlative protection. But is it the correlative protection? IgA is really important to look at, and T-cell repertoire is really important to look at, because I can't emphasize enough that knowing something is better than nothing. But actually, all the stories of viral immunity that have been worked out have been quite nuanced and quite different, and the devil has been in the detail.
Definitely. Thank you, Danny. So we had some questions around some of the testing, and one of them that came in is, are you aware of any B cell ELISpots, ELISpot, that are available to detect early antibody response?
So I've been trying to keep quite a close eye on what people have been doing and telling me. I haven't come across any examples yet, but I bet there are examples out there. So again, it comes back to the kind of thing we were talking about before, doesn't it? That really, really important to know in terms of both the specificity and kinetics and and scale of that response, and doable in many kind of research labs or hospital labs, but not at the kind of scale that people wanted at the moment.
Great, thank you. Definitely a watch this space then, isn't it?
Yeah.
And there are a couple of questions that came in about the innate immune response, as you would imagine, Danny. So what role do you think the innate immune response plays in viral detection and in the subsequent cytokine storm?
Absolutely enormous and important. So there's some clues out there already, aren't there, which are slightly reminiscent of other stories in coronavirus infection and slightly reminiscent of influenza stories in terms of initial signaling and pathways, things like the IFIT pathway and type 1 interferons. Some evidence that this virus may have some ability to subvert some of those pathways. Probably early days in collecting data on whether genetic polymorphisms matter for your susceptibility through those pathways. So enormously important, I think, both in that initial switch for, am I going to be an asymptomatic person who didn't even know I had the infection, or am I going to be one of the severe symptomatic cases? And also on the sort of negative side for, am I going to be one of those people with, with massive IL-6 levels who ends up on ICU?
Yeah, and you mentioned. Oh,
no, carry on.
I know. So you mentioned influenza, and I mean, it's from the data that's come out so far. What levels of parallel do you think we can draw between SARS-CoV-2 and influenza?
So, I think it's kind of been helpful up to a very sort of minor point, and therefore, and afterwards, slightly unhelpful, hasn't it? Because beyond saying kind of, very broad brushstroke truisms like, these are both very severe respiratory pneumonia-inducing pathogens with a high level of immunopathology. Beyond that, I think it's been kind of unhelpful to try and draw parallels with influenza, and has possibly even led people astray, I think. Beyond the broad brushstroke.
No, definitely. And then we had some questions in about vaccine development. One of them being, what, in your opinion, what are the key aspects that need to be considered when it comes to in silico prediction of T cell epitopes for vaccine development? So,
I think a lot of the established pipelines have gone into this and will go into this, and that includes an element of antigen discovery, antigen prediction, and epitope prediction. And the point I was trying to get across when I showed the slides at the end are that on the one hand, immunologists and vaccinologists should really sort of pat themselves on the back that so much is known, and people thought the response to Ebola was really fast, but much faster, and even more stupendous. And yet, there's many a slip on the way. Things that look promising in terms of inducing immunity in the end don't turn out to be quite the real thing in terms of protection. So you need a number of candidates, a number of approaches, and just work out empirically which one works best.
Mm-hmm. And in terms of the ongoing vaccine development, I mean, you ended with a hopeful message. I mean, what's your views on what we know so far? There are 20 or 30 clinical studies on the go at the moment, aren't there, globally? But what's your take on where we are in that vaccine development process?
I just think that people who watch this space have learned over the years to adopt a kind of kind of careful, cautious optimism that I think there are some terrific people in this space doing brilliant work using kind of tried and tested approaches. We have faith that they can work. We have faith that they have worked in the past, and yet the people doing it are canny enough to know that we've got to have a really careful eye on the details of which are going to be the best candidates and which things are really, really going to work and be protective, and do that in a way that's compatible with rolling out doses on the kind of scale and quality that we're going to need. It's a really, really tall order. It's like a moon mission.
Thank you, and I mean it's warming to know that the global community is pulling together and carrying out these studies, which is fantastic. So changing topics slightly, moving more into the zoological space, do you know if bats have neutralizing antibodies against SARS-CoV-2?
I've been trying to study the papers. I don't think I've ever seen a paper on that, so I will have a look afterwards. I'd love to know. So obviously I've seen lots of sequencing papers about the bat viruses. I've never seen any antibody papers. I'd love to know. I don't know.
Well, when you find out, please do let us know, and we'll pass that knowledge around.
Yeah, we'll check.
And then we've had some questions around the antibody antigen testing, and in your opinion, how close do you think we are to being able to test with good specificity for past infection and immunity?
Yeah, so I was trying to kind of convey the kind of complexity of it, that it's not my territory to talk about the kind of public health or political aspects of that, because that's way outside my remit. So all I can comment on is the immunology, and say that if you wanted in a hospital or research setting to be able to characterize hundreds or thousands or even tens of thousands of serum samples, you could do it next week. That isn't necessarily going to get you to the level you want to be at on a sort of national or worldwide basis. So I accept that one might need some kind of combination of approaches between the sort of hospital lab-based testing with with the sophistication of a lab and all its kit and plate readers and things, and some further tweaking of those commercial modules over the coming period to get that really sort of wide scale rollout. And I think there's a place for both of those things.
I'll mute myself. Brilliant. Thank you, Danny. And I think, Danny, we should draw it to a close there. We've got to our time here of 12:15, but that was a fantastic talk, and thank you for your views and your take on the research that's been coming through in the last couple of months, but also on the huge number of questions that we've had in. They're still coming in. So thank you everyone for your engagement as well. Really, really appreciate it. Just for your information, we are now recording these webinars, and we will be making them available on our website. So the one that Arnie Akbar gave last week will be on our website soon. And hopefully in the next couple of days, this one from Danny will be on the website soon. So if you want to loop back and listen to it again or share it with your colleagues, please do go ahead and do that. If you could spare 1 minute of your time, we had a great response to the feedback questionnaire at the end of the last webinar. If you could spare another minute of your time, or a minute if you weren't here last week, just to fill out that questionnaire, it really will help us improve these webinars as we go on through the weeks and we having different speakers. So please do give us your views on that questionnaire. And as we've said, there are more webinars to come. We will have Danny Altmann up again, giving us some really great overviews of the emerging research. And we've got a whole host of other speakers that we're in the process of, we've confirmed, but we're in the process of getting dates for. And these are speakers not just in the UK, but global leaders in other countries as well. Please do keep an eye on the emails that we send you and on our website and on Twitter. That is how we will be promoting these webinars. And don't forget, the British Society for Immunology is here for you. So please do find out more about us on our website. Do follow us on Twitter, BritSocIm, im. And if you want to get involved, then please do contact us and join us as an organisation, but also get involved in the activities that we are delivering. So that just leaves me to thank you again, Danny. That was absolutely wonderful. And thank all of you who have signed in live today. Really great to have you join us. And we look forward to seeing you next time. Thank you, everyone.
Thank you.